intertwine perfectly. They're all right where they should be. A is paired up with T, and C goes with G. You know the shape of DNA is a lot like a letter. A lot like a letter.
Let's take a look at things from the level of a cell. So I'm going through the cytoplasm, passing all the organelles. Look inside the nucleus, which is the cell's brain, and you'll see some floating molecules of DNA. It's got a funny shape, double helix is its name. Has genetic information, so we all don't look the same. Has a phosphate and a sugar called deoxyribose, and nitrogenous bases that are lined up in rows. DNA gives us traits from the unique genes, but its other main purpose is to make proteins. It really is important, it's essential to our lives. Without DNA, we couldn't possibly survive.
Nucleotides intertwine perfectly. They're all right where they should be. A is paired up with T, and C goes with G. You know the shape of DNA is a lot like a letter. A lot like a letter.
We're always learning new things about DNA. Gregorman Delta innovators of today. The first model was made by Watson and Crick, but their idea was stolen from a girl, Rosalind, who was taking some pictures with her fancy x-rays and discovered the structure of DNA. But before she could even put her findings to the test, Watson and Crick up and stole it off her desk. But they weren't the only ones who made a contribution. Shargoff also came up with a solution. He said A goes with T and C goes with G. G-A-R-R-E-T-T.
We need our DNA to stay alive, but we would run out if it didn't divide. In a process called DNA replication, the enzyme helicase acts as the initiation by splitting apart the two strands of DNA, making a replication fork. The source will replicate DNA polymerase, carries two nucleotides, binds them to each strand so the cell can divide.
Nucleotides intertwine perfectly. They're all right where they should be. A is paired up with T, and C goes with G. You know the shape of DNA is a lot like a letter. A lot like a letter.